Good afternoon and welcome to NRGene's webinar series. We are Gallant and Fna from the marketing team. Today we will be discussing an innovative genotyping solution, a new product called ArrayMAGIC. We will save time for questions at the end of the webinar, and we encourage you to send us your questions during the presentation using the question tab. This webinar is also recorded and will be posted on our website in case you want to give it another listen. We have two excellent speakers to guide you through today's webinar. The first is Dr. Paul Comet, NRGene Senior Technical Specialist. Paul has extensive experience in molecular breeding and biotechnology with over 25 years industry experience and a PhD in genetics. He will share some insights about genotyping and about our new product, in particular, NRGene's ArrayMAGIC. He's joined by our guest, Dr. Mark Gilman, a professor of plant molecular biology at Penn State University. Mark has a PhD in genetics and deep experience with molecular breeding tools and biotechnology. He currently studies crop improvement while teaching the next generation of scientists about plant genomics and genetics. We hope their insight in this webinar will be useful for commercial and academic breeders, molecular biologists, and genetics who are looking for high throughput, high quality, low cost genotyping solution.
Thanks for the introduction, Daphne. It's great to have you join us today, Mark.
Thanks a lot, Paul. It's great to be here. As you know, I work on cacao and we're just getting started in this domain, so I'm looking forward to learning how my lab might use the technology you're going to present in the future and how it could be of value for everyone joining us for today's webinar. Maybe we could just start with a few words about NRGene and what we could expect over the next half hour.
Hi guys, this is Gal from NRGene, and I'm going to be speaking to you about a little bit of background about NRGene. So we're going to start this webinar by describing a little bit of who we are and how we got started. Then we'll introduce genotyping applications and existing genotyping solutions. We'll also provide an introduction to our new genotyping product, which utilizes our haplotype databases, and we'll explain some of its benefits. So as for who we are, NRGene was established in Israel in 2010. Our initial range of products, including our genome assembly services, were released to the public at around 2014. Now we have a full set of tools for breeders that could reduce time and improve efficiency throughout the breeding process. Some of you probably know NRGene for our genome assemblies. We've delivered hundreds of assemblies for our customers, but we've also delivered unique analytics capabilities to some of the leading agriculture companies such as Bayer, Syngenta, and non-agriculture companies like Bridgestone Tires, Toyota, and more. We look to offer our services to advanced breeding and product development across a wide range of industries, academia, government agencies, and nonprofits. Recently, in late 2018, we've opened our US office in San Diego, California, extending our global outreach with other offices around the world. So the question is basically how it all connects. NRGene has a number of products that fit together as a complete solution for different molecular breeding programs in plants and animals. Right now, building up robust molecular breeding capabilities usually requires a physical map, a reference genome. This is our de novo MAGIC product. Performing a detailed comparative analysis to understand diversity, genomic changes, or structural variations requires a comparison of genome sequences. Our PanMAGIC service does just that. From multiple assemblies, we can generate an understanding of the variation genome to genome and provide you with the ability to position between and visualize the multiple genomes. We've also offered our trait services and launched it recently, ArrayMAGIC services, which include quantitative trait locus or QTL mapping and the delivery of trait-associated sequence markers. Essentially, this connects the dots between molecular markers such as SNPs and observed traits. The ArrayMAGIC system holds diversity information about a species as sequence-based haplotypes. As for a new product, ArrayMAGIC, this is a genotyping-based system. This is a genotyping system based on ultra-low sequence coverage. It utilizes our sequence database and our unique analytics to deliver very high-density genotype calls in those species.
Basically, to clarify what Gal was presenting, you're saying that the ArrayMAGIC genotyping service is not really done with an array, and it's a sequence-based technology. And I also found on your website, it also says it's a novel approach. Can you put the ArrayMAGIC service in the context of other genotyping methods?
That's a great question, Mark. I'll let Paul take it from here.
Yeah. So we can list some common marker applications in the field we work in. And as you know, there are a number of technologies that can be applied in each of these applications. So, why do you genotype? And here's a list of these applications. A lot of the markers goes into marker-aided selection, marker-aided backcrossing, and genome selection. And each technology has a particular niche, as there's a lot of factors to consider. For example, the number of markers per sample and the distribution of those markers around the genome. In all these cases, you're likely weighing cost, information content, and ease of use. You have to consider not just the per sample cost, but also the cost to set up the system. And so, information content is key. In a simple segregating population, you want to balance the marker density with the recombination frequency in the population. But if you want to develop trait-associated markers, or you need to consider things like the haplotype structure or the makeup of your parent germplasm, you're going to need high-density marker information. And in addition, you don't want those markers that you're using to understand this diversity to be biased by methods you use to discover the markers in the first place. Of course, missing data and error rates are also important. Now, there are a number of technologies that have advantages and disadvantages based on the parameters I just outlined. Here, in this slide, I categorize marker density according to the technology limits or the practical limits of the system. So, here you can see single marker assay down to genotyping by sequence. Does that make sense in relation to what your work is, Mark? Mark, do you guys hear me?
Yes,
Sorry
we
about
can
that.
hear.
Yeah, sorry, Paul. Yeah, that does make sense. And I wanted to say this is a great case where in my favorite species, cacao, we're just now developing trait markers. And the single marker assays will be extremely useful once we have those key traits mapped. But to do that initial mapping, we'll be developing a good set of genome-wide markers. And I think we'll want to genotype with a technology meant for high densities like you list here.
Yeah, so I mean, in a perfect world, it would be great to have a very inexpensive technology that's flexible with little or no setup cost, with high accuracy, high information content, fast and simple to use. This would replace, many of these technologies that I list. Sequencing technology has definitely moved the field closer to this ultimate solution.
Mm-hmm.
So when we look across technologies at estimated cost per sample versus marker density, you can see that the common technologies listed here are quite broadly distributed across the marker density spectrum. And up at the top, I indicated the applications usually used for these densities. You'll notice that no technology really spans across the applications that I talk about up here. And so that's where that ultimate solution would be really useful. Today's ArrayMAGIC product offering is at a very high density, as indicated by the circle. So that black circle indicates what we're delivering as a product. And this is by our choosing. But due to how we do this, our technology, it's possible to deliver higher or lower density information as indicated by that dotted line, the black dotted line.
Right.
I'm very interested in hearing, from the audience later on or through emails as to the most useful output of data for their needs, whether it's lower density or higher density. But right now, like I said, the product is focused on giving a set of SNPs that we'll talk about later at very high density. So please give me some feedback as we go. As you mentioned, Mark, when it comes to applications such as mapping or marker discovery, having the higher density genome-wide sequence-based markers would improve the outcome of all of the downstream work associated with it.
Yeah, I think that's true. Most people would take one comprehensive solution over a bunch of more niche tools that have certain downsides. And NRGene's ArrayMAGIC definitely fits into the sequence-based approach. But one question I think we really haven't answered yet, however, is how does ArrayMAGIC do that, and what gives the technology its edge?
That is a key question, and I want to try and explain that. NRGene's approach is different for several reasons. As we noted earlier, the standard technique relies on single reference genome and have a potentially biased approach to marker discovery based only on the SNP information and that reference genome. To move beyond this bottleneck, NRGene's solution is built with a pan-genome database of diverse reference grade genomes. And then positioned into that pan-genome structure is a diverse set of haplotype sequences. So long sequences fit into the pan-genome. As you know, the genomes of organisms are transmitted essentially as chromosome segments, or things we call haplotypes, and these haplotypes can be defined by sequences. And so in this way, we capture and position haplotype sequence information rather than just SNPs. So the database holds much of that unique sequence making up the population. The utility here is to efficiently and accurately impute or call high-density markers, as well as the discovery of new markers, which dynamically improves the database as we go forward. So let's take a very simple analogy to describe this genotyping calling capability of this NRGene system, okay? So imagine NRGene's database as a library where we have every edition and version of a book in this. And in this example we're going to go through, I'm going to use Romeo and Juliet. So in each book, the sentences hold all the important information. And of course, each edition has the same plot and content, just like genomes of a species. However, each edition has sentences with relatively small changes in the text organization, like sequence polymorphisms. And so we're outlining that in the colors at the top. So in this example, our database holds 3 versions you see at the top with the text variations outlined. If we were to skim the words or skim sequence, we would identify a few key letters in each sample. Because the database holds the complete sentences, we can impute or correctly identify not only the rest of the sentence, but also the version and the position from which that sentence was derived, thereby identifying the makeup of your book, your sample.
Wow, that's a great example how you can get back to such high density information from the database. It helps me understand it a lot better. If I was a customer for the service, how would it actually work?
Yeah, so NRGene has developed an ultra-low sequence workflow, and it's outlined here. It taps into NRGene's proprietary databases, so the users can make these genotype calls from such data. Right now, we're offering genotyping for maize and soy. Some key crops like cotton, tomato, and canola are going to be available in the near future. The technology can be applied to just about any other animal or crop with the appropriate database that we build. After you go to NRGene's website and fill out a request, we'll send a leaf sampling kit, and then your team can use it to collect leaf tissue punches from a population of related plants, along with the parent tissue of that population. Alternatively, you can supply the appropriate amount of DNA instead of tissue. And then NRGene works with sequencing providers to generate those libraries and sequence data, that ultra-low skin sequence data. The data then is uploaded to a cloud server where NRGene's analytics performs the genotyping calls. So then as customers, you retrieve a VCF file from a secure site when the data is complete.
So really all a customer has to do is send in the tissue sample and NRGene will take care of the rest.
Exactly. That's exactly it. And then, behind the scenes, the genotyping is performed using this unique library prep and Illumina sequence data to produce about a 0.01x coverage of the genome of the progeny, of each one of those progeny. So, for example, for maize, it comes to about 25 megabases of sequence data per sample. The skim-seq reads are compared back to the database, which holds those unique sequences that define each haplotype, including the parents of your population. Once a string of sequences is identified, we then infer or impute the rest of the sequence within the haplotype region. Because the database holds that high resolution sequence information, just like the example of the book information, we rationalized that the output of the system would be the genotype call at a known set of markers that researchers may be familiar with. So for maize and soy, we are reporting out on the union of Affymetrix axiom array, set of SNPs, as well as the standard Illumina array. This amounts to about 210,000 markers in soy and about 618,000 markers in maize. Alternatively, we can input a low density set of genotype values for the population and impute that to a higher density instead of actually doing the sequencing and genotyping. So why would you want this service to bring a low density set of markers up? Well, I can tell you there are times in experiments in the past for me when I had a QTL mapping database or data set that I wanted to integrate with additional experimental data. But the genotyping was done with different marker sets or was a different set at a lower density. Using this product, using our SNP imputation service, you just need to provide the progeny SNP data instead of the tissue, along with the parent tissue or sequence. And so from there, we return high quality and high density imputed SNP data that reflects this higher resolution information that's in the parents. This means you can then integrate these data sets using common genotype data and data that is much higher resolution than you had before.
Okay, so it sounds like the ability to make a genotype calls is really enabled by the deep sequence information that you hold in the database. But with such low sequence coverage, how much data will be missing and how accurate can it be?
Yeah, good question. So we actually did a series of tests in Maze using 4 populations with parent data. With the 600K axiom array, in the literature, it was reported that you get back an average of about 95, 96% of the markers per sample. So a normal straight up axiom array genotyping, you get about 95% return. Okay, now in our system on the left, you can see the answer to both of your questions. At 0.01x coverage in Maze, we return a call of over 578,000 markers. That's 98% of the axiom call rate. And on top of that, the error rate was determined to be below 0.082%, really highly accurate. You can see on the right now for the SNP imputation that if we use the system to impute from 300 or 3,000 SNPs, the part that's circled, we can return over 513,000 markers, and the error rate there is below 0.2%. Now, we did this test also in soy, and we get very similar results, returning close to 200,000 markers when we do the skim sequence at 0.01x, and again, very low error rate. Now, obviously, there's a lot more to this product that we can cover in this short webinar. So what I encourage you to do is head to our website, www.nrgene.com, to explore these features and benefits a bit more. There's a lot more information on the website as well. And you'll find that we're offering a trial package at the moment, starting from 96 samples per order, to give this a try and see how it works with your data in your hands. So just to sum up what we went over today, I want to make sure I communicated, we all communicated with the number of us talking, that we have a turnkey genotyping service in maize and soy that returns accurate, complete, very high density genotyping for a very competitive cost. We can output the VCF file. So this is kind of important. We output the VCF files so the marker positioning is relative to any of the NRGene available reference genomes. And you can find those on Maze GDB, on the Maze genetics database.
Well, that's really cool. Thanks, Paul, for walking us through this interesting and unique approach to sequence-based genotyping. It really looks like a very efficient method to get at high density marker data and a real breakthrough. I believe Daphne is now signaling that she's receiving some questions online or being sent in, and I was wondering if you could just take a few of those for us.
Yeah, let's take a few questions now, and I'll certainly answer any additional questions by email as soon as possible.
Okay, so first question we got is, what happens if I don't have the parents of the population?
Yeah, so as I tried to explain, and I know we have to go through this more to really get into the details, but the product is built to give maximal accuracy with this minimal cost, relying on the higher call rate of the parental samples. So we use that parental data. If your parent lines or their progenies can, or the progenies can be found in our collection, in our database, we can impute based on those instead of your original samples. But you have to make sure and take notice that differences between your germplasm and the line in our database are going to be overlooked in cases like this. So accuracy might change.
Okay, and another question that we got is, will you retain my data in your database for me to use, for others to use?
Yeah, that's a great question. You know, the data is completely yours. Oh, and we'll return it for a fixed, we're going to retain that for a fixed period of time in the database to do the calculations, and we'll discard it unless you specify to us to keep it. As the data is very useful for reiterations of genotyping services, whether it's with you or others, We highly recommend to keep it in the database, but we leave that up to the customer.
Okay, now another question. What is the time estimation from DNA to receiving the file?
Yep. So our turnaround time is 8 weeks. But please, as you go in and try this out, please discuss your timelines, your needs, and your project with an NRGene representative when you're ordering this. Okay, so that's important to do. We want to hear what your needs are, and we want to get your feedback.
Okay, thank you, Paul. I'm sorry, but we couldn't answer all your questions, but please note that we will answer them all in writing right after the webinar. Thank you very much for joining us today. Enjoy the rest of your day. Bye,
Thank you,
Paul.
everyone.
Bye, Paul. Thank you.
Bye.
Bye-bye. Thanks, everyone.